This new video is about cell division, and the first part of it will cover mitosis and binary fission, while the part B will cover meiosis. Cell division in this part of the chapter will in fact give rise to cells that are identical to the cell that produced them initially. So it is meant to reproduce cells in a colony or to make more cells in a growing or developing organism, as well as in order to renew the tissues. So regularly we need to replace those cells that died or that were damaged because of just the metabolic activities associated with the life of an organism. So all this will need to reproduce cells, and this is what will happen during cell division. So the goal of cell division is to reproduce cells in order to enlarge the population. So as we said, in a colony or in a tissue or in an organ. So it will be to reproduce cells that are identical. The challenges that it will face will be, first of all, to separate the genetic material equally without any gain or loss of material when you compare the cell that divided itself into two daughter cells. And it will also be to separate the cellular content equally, so you don't want to have one of the cells that takes everything while the other one has almost nothing. So therefore, cell division is a very tightly regulated process that occurs in several steps and in a very regulated fashion, so that everything occurs accordingly to what we just said, so that you don't lose or gain material in any part of the cells. So one mother cell, when it divides, will give rise to two daughter cells. So you don't have anymore a mother cell at the end of the process, because it became two daughter cells. These two daughter cells can eventually become later mother cells themselves and give rise to again two daughter cells each, so four in total. So we have a little joke, but that is pretty true. It is that in fact in biology it is the only science where multiplication and division mean the same thing. So when a cell divides, you will in fact multiply the number of daughter cells, and this is what is shown in this little picture. So I said that cell division is a tightly regulated process. However, it doesn't occur exactly in the same fashion for prokaryotes and eukaryotes. So first of all, you remember probably that one of the main difference between prokaryotes and eukaryotes is the presence of a nucleus in eukaryotes only, while prokaryotes don't have a membrane enclosed nucleus. So it will slightly change the way the division of the genetic material will occur. The genome is also slightly different, because in prokaryotes you have only one circular chromosome, while in eukaryotes we have n linear chromosomes, so a certain number of linear chromosomes, but a number that is different from one. And DNA, however, is the same in prokaryotes and eukaryotes, so I decided to write that again just because it is a very common misconception. Remember that both prokaryotes and eukaryotes have double-stranded DNA, and nothing has single-stranded DNA, okay? One unique double-stranded DNA makes a single chromosome, but it is still a double-stranded DNA. Then, how do we call the cell division process? In prokaryotes, it is called binary fission, and in eukaryotes, it is called mitosis and cytokinesis. So we will first review the binary fission, and in a couple of slides, we will cover mitosis and cytokinesis. So here is the process, the general process of binary fission. So it is a process that occurs almost only in prokaryotes. You will, however, also see that in some organelles of eukaryotic cells, for example, mitochondria, reproduce themselves by binary fission. So the first step is that the chromosome, the unique chromosome of the bacterium, is attached. So the DNA molecule will be attached to the inner side of the membrane. and this spot will also serve as an origin of replication. So the replication of DNA, or the copy of the DNA, will start at this specific location, and it will progress bidirectionally around, like, from the two sides of the origin of replication. So if you see here, it is one of the sides, and there is an opposite side where the replication progress. So the circle will be reproduced identically, and the two origins of replication that have been replicated at the very beginning will both remain attached to the membrane, but a synthesis of membrane will occur between the two spots so that the cell elongates, it grows in length, and because the membrane is produced between the two spots where the DNA molecules are attached, it also brings with it the two molecules of DNA, both sides of the cell. Once they are far enough to be separated, then a membrane will form between the two DNA molecules so that you can now form two daughter cells, and each of these daughter cells can start again step 1 and another round of binary fission. So in bacteria, this process is relatively uninterrupted. As long as the bacteria have enough nutrients and all that, they will continuously go on from step 1 to step 6, and then from step 6 to step 1. So this is how a colony of bacteria can grow pretty fast. Some of them can grow as fast as double the population every 20 minutes. So this is it for the binary fission. You see here an example of, in fact, a real picture of bacteria dividing by binary fission. So now let's have a look on the eukaryotic cell cycle. So this is the cell cycle that will be seen in all eukaryotic cells, so including the unicellular cells that will make colonies, as well as multicellular organisms in which cells will reproduce in order to make tissues and organs. So this is a tightly regulated process, as we said previously. It will include 4 different phases and 4 different events. So first, it will involve the growth of the cytoplasm and the reproduction of organelles. This will occur in a phase called interphase. The replication of the DNA, or the copy of the DNA, so again it occurs at the end of the interphase. Then the division of the genetic material that has been recopied in the previous step. This will occur during mitosis, and the division of the cytoplasmic material, which occurs in the final step, which is the cytokinesis. If we look at this in a more visual fashion, so you see here on this picture that represents the cell cycle that the interphase is the biggest part, and the M phase, which includes the mitosis and cytokinesis, is the smallest part of the cell cycle. Now, we have to differentiate cells that are actively reproducing. So, for example, cells that are in an embryo and the embryo is actively growing, and you need to populate this growing organism with new cells. So there is an almost uninterrupted succession of interphase and M phase. So cells are constantly dividing in order to make this new organism grow. It is also the case, for example, when you had a disease that destroyed an organ like the liver, it will be able to regrow after a phase of active reproduction of the cells. So in this case, you will have an almost uninterrupted succession of these interphase and M phases. But for most cells of an adult or even an already relatively well-developed organism, you will have a different phase that is in between the two, and it is called the G0 phase. It is a phase where the cells are not dividing, so they are not in a phase of the cell cycle. They're not in G1, they're not starting to grow a cell in order to divide it. They are simply in a phase of differentiation, so they are out of the cell cycle. And in fact, most of your body cells at all time are in G0. They are not dividing, they are just working, differentiated and working. So for example, a cell of the liver will make liver enzymes, a cell of the hair will make keratin, and they will not be dividing at that time. Now, when cells are dividing, you have to remember that one round of cell division will always be made of one interphase, which includes a G1, S, and G2 phase, and the M phase that includes mitosis and cytokinesis. So we will review these two parts, interphase and M phase, successively. So first have a look at the interphase. Interphase is itself divided in three steps, and the three steps are called G1, S, and G2. The first one, G1, has been called like that because initially when researchers looked at cell division, they observed it in a frog embryo, and they noticed that there were moments where they couldn't see any sort of activity, any sort of visible activity. So they called these periods of times gaps. And after that, with new techniques and improved microscopy, we could realize that in fact it's not really a gap, it is rather a phase where the cell actually does a lot of things. However, we cannot visualize them very easily, so this is why they called it gap 1, but now we tend more to call them growth 1. You can use both words. In any case, it is G1. So G1 is the phase where the cell will grow, where it will synthesize more membranes so that you can eventually make two cells with the existing cell. So you need to have an increased amount of plasma membrane, internal membranes as well. You will also reproduce organelles so that the new cells will have enough organelles. Proteins will be synthesized as well so that you don't lack those proteins that are needed for the replication of DNA. And at the very end of the G1 phase, the DNA integrity will be controlled, checked, and in order to make sure that there is no damage, mistake, or break, or whatever that could impair with the copy of the DNA, or that could eventually reproduce a mistake in the cells soon to be formed. Once this is all done, we can switch to the next phase that is called the S phase. S for synthesis. It is the synthesis of DNA. Basically, the DNA of the mother cell will be taken as a template and copied into a newly synthesized DNA. So it is an exact copy that will be made, and one identical copy of each chromosome will be produced during the S phase. We will now have a closer look at the S phase and what happens to the genetic material at that time. We won't cover here the replication of the DNA at the molecular level. It will be covered in a different video, but let's look at the chromosomes and what happens with them. You all remember that in eukaryotic cells, most of the eukaryotic cells contain two copies of each chromosome. They are diploid. So the very first cell of a diploid organism, like the embryo, receives one copy of each chromosome from one of the parents. So here on the pair of chromosome that is represented on this picture, you can imagine that it is, let's say, chromosome 1. So one copy is received from one parent, and the second chromosome 1 is received from the second parent. This is a pair of homologous chromosomes. So for most of the cell's life, the chromosomes, even the homologous chromosomes, will be found exactly like it is shown in the picture. So it is a kind of stick-like structure with one single piece of DNA. But remember that every piece of DNA is made of a double helix. So it is a helix made of two strands of DNA twisted together. So each of these sticks here is one piece of DNA. What will happen during the replication of the DNA is that the two strands will untwist, they will open, they will become separated, and each of them will serve as a template to synthesize a new strand on each of them. So basically, what will happen is that from one piece of double-stranded DNA like you had initially, after replication of the DNA, you will end up with two pieces of DNA that are the exact same copies of each other. These two pieces of DNA will stay connected together by the means of a part called the centromere, and they are called sister chromatids. So sister chromatids are the two pieces of DNA connected by the centromere, and it is not to be confused with homologous chromosomes that are two different chromosomes of a same pair. Like here, the dark blue and the light blue. If we look at what happens genetically, I want you to figure out what exactly occurs with the genes. You know that the chromosomes contain, in fact, are made of a piece of DNA. The piece of DNA in question is a sequence of nucleotides that are attached in a specific order, and this order codes for the production of proteins in your cells. And the codes are called genes. So if you look at a gene, for example, that was located on one of the parental chromosomes. So let's say the dark blue is inherited from your father and the light blue is inherited from your mother. They contain the exact same genes, okay, at the exact same locations. So if we imagine that the gene represented here codes from the color of your eyes. So you inherited one gene from the father and one gene from the mother. They are the exact same sequences with just slight differences that can make the color different. Okay, so for example, blue or brown or black, whatever. When the S phase has occurred and the DNA has been replicated, each one of these genes will be copied exactly in the same fashion. So the sister chromatids of the father's chromosome of the homologous pair will contain the two genes encoding for the same color. And the two sister chromatids of the chromosome coming from the mother will contain the same gene as the initial chromosome but copied twice. So it is again the same gene as the one inherited by the father. It codes for the color. Okay, it's the color gene. However, instead of a black color, it is a blue color, for example. So once this is done, we have copied the genetic material at the identical, so it is the exact same thing that is just found in two copies. Okay, again, if you look at the number of genes encoding the color of the eyes, you have 2 at the beginning and you have 4 at the end. After that, we will enter in the G2 phase. So the G2 is the second gap or growth 2 phase. It is, as the name indicates, the second phase of growth. It is a phase where the synthesis of proteins needed for mitosis will occur, and it is a phase also where you will control the DNA integrity, because when DNA is copied, it is opened, so it is a bit more sensitive to potential damages. It is also subject to mistakes during the replication process, so all that will be fixed and repaired during the G2 phase. And another point of control occurs at the end of the G2 phase, but if everything goes well, then the cell will enter in the M phase. It is the mitosis and cytokinesis phase. So during the M phase, the first part will be mitosis that will be needed to separate the genetic material that has been copied in the previous phase during the S phase of interphase. And after that, we will have the cytokinesis, which will separate the cytoplasmic material, but we will see that a bit later. So let's first review mitosis. Mitosis is a succession of very well and tightly controlled steps. In total, we have 5 steps in the mitosis, and they are called prophase, prometaphase, metaphase, anaphase, and telophase. Each of these steps has a specific function, and it is very important that each of these steps is completed correctly before you go to the next one. So if we have a closer look at mitosis, first you remember that at the end of the G2 phase, we have our DNA completely replicated, and it is under the form called chromatin, so it means that it is associated with proteins, but you see that it's completely intermingled and not at all condensed, so you will need to condense that before you enter in the division, really, the real division of the genetic material. In the G2 phase, we also still have our nuclear envelope that is well formed, and at the level of the cytoplasm, we have centrosomes, which are structures made of proteins that include a centriole pair, and they will be the point of start of little microtubules that will make a spindle and help with the division process. So at the end of the G2 phase, if everything is made correctly, we can enter into the prophase. So the prophase itself, here shown in the middle picture, is where at the nuclear level you will have a condensation of the chromosomes. So what was previously a piece of DNA duplicated, long and completely intermingled will now start to be condensed. So you will start to be able to visualize the sister chromatids of each chromosome connected by their centromeres as highlighted here. And at the level of the cytoplasm, you will see that each centrosome will grow microtubules that will push the other centrosome away from the first one. So it will start to synthesize what we call the early mitotic spindle, where each centrosome grows microtubules in facing each other so that they will move apart. So also at this step, a centrosome containing growing microtubules is called an ester. Then once this is made and completed, we will enter in the prometaphase step. So this is the step at which you can start to really visualize very well the chromosomes. So what happens at the level of the nucleus is that first of all, you see the nuclear envelope that starts to dissolve and be removed from the cell. And at the same time, you see the chromosomes that start to attach themselves on the growing microtubules of the now almost completely made mitotic spindle. And they attach by the mean of a structure called, in fact, by the mean of their centromeres, but in a structure called the kinetochore. The kinetochore is the structure that attaches the chromosome to the microtubules. And the microtubules that do contain kinetochores and chromosomes are called kinetochore microtubules, while the ones that don't contain any chromosomes are called non-kinetochore microtubules, and they're used to help with the alignment of the mitotic spindle and the growth of the mitotic spindle. So these growing microtubules are able to go in the middle of the cell because the nuclear envelope has now been dissolved and all the chromosomes can merge in the can join at the center of the cell. So the next step is the metaphase, and during the metaphase, the first thing that we can notice is that the spindle is completely formed, with the microtubules spanning the entire middle of the cell, while the centrosomes are each found at a pole of the spindle. The chromosomes, or the genetic material, they are still attached by the means of their kinetochore on the microtubules, but they all align at the center of the cell in a plane. Remember that this is in 3D, so it's not in a line, it is in a plane, and this plane is called the metaphase plate. At this step, you are almost ready to separate the material, and this is exactly what will happen during the next phase called the anaphase. In fact, the centromeres of each chromosome will separate, it will be cleaved, and during the anaphase, each side of the centromere will be pulled by the means of the half kinetochore on its half microtubule, on its side of the cell. So at this time, instead of calling them sister chromatids connected by one centromere, you now call them daughter chromosomes because they are fully individual chromosomes at this step. So they will move progressively towards the centrosomes, or the poles of the cell, and this will separate all the genetic material equally so that each side of the cell will receive one copy of one of the sister chromatid made during the S phase. Now the last step of the mitosis is called the telophase. So during the telophase, what happens is that the chromosomes start to decondense, so you will see them coming back to their initial state where they were made of a single strand of chromatid that was very intermingled to the other chromosomes. And you will see that the microtubules have now regressed, and the two centrosomes, are now found at each extremities, but you don't have anymore the mitotic spindle. And at the same time, the nuclear envelope will reform so that the chromosomes will be again enclosed in the nuclear envelope like it was previously. So we're almost done with the two daughter cells. They're, however, not fully separated yet. This is what will happen in the next step, that is called the cytokinesis. It is the step where the two cytoplasmic compartments will be totally separated. So this occurs differently in animal cells from in plant cells. So in animal cells, what will happen is that cytoskeletal filaments, the filaments will form a ring that is parallel to the mitotic spindle. And even if the mitotic spindle has now disappeared, the ring is parallel to the orientation of the mitotic spindle. The ring is attached to the membrane, the plasma membrane. So as the ring contracts, it will pull the membrane, and this will create a cleavage furrow that will totally close itself and separate the two membranes in two daughter cells. In plant cells, things are slightly different because the cell wall is too rigid to be pulled at by a cytoskeletal ring. So instead, what will happen is that vesicles made of a membrane very similar to the plasma membrane will form in the middle, in the plane between the two nuclei. And these vesicles will fuse together in order to form a full plate that will eventually close the two compartments. And at the end, a new cell wall will be formed between the two membranes of the two daughter cells. So this closes the chapter on cell division.